so it will be diffusing out. Okay, so I think your screen is lagging. Can you try to restart your internet connection or close all tabs so that it works efficiently? Okay, so this is the basic process. Okay, can everybody understand? Is it much better? Any questions? All right, awesome. If there's if don't understand, do raise a question. Okay, it's important that you come into the study session and start to ask. Can I mention negative pressure in transpiration pool? Negative pressure. Uh, can. Okay, so repeat adhesion and cohesion. Cohesion is between one water molecule with another water molecule. Okay, refer to this diagram. So cohesive force, okay, which is cohesion, is between one water molecule with another water molecule. Do you understand that? And adhesion is between one water molecule with the wall of the xylem. Okay? Is this making more sense now? All right. So there's no question, then we will proceed. Okay, we'll do some sample question. So the first question is predict and explain the effect of high humidity on the stomata. All right. When they come to this kind of question where they ask you to predict, it wants you to make sure that you understand what is happening. So this question, when they mention high humidity, is referring to the factors that affect the rate of transpiration. So factors that affect the rate of transpiration. Okay, so one of the three factors, of course, is temperature, humidity, and also wind speed. So since the question is now saying high humidity, in optimal condition, in order for transpiration to occur, it must have a lower humidity so that the rate of transpiration is high. So now this question is saying what happens at the higher humidity? Yes, correct. So the rate of transpiration will decrease. Transpiration will decrease. Okay, you satisfy this part of the question now. Now the question wants you to explain the effect on the stomata. So this question you cannot simply memorize and then vomit out into this question. You will confirm fall into the trap. So how are you going to start answering this question? So when you come to this point of answering that is referring to the stomata, right? We need to see how the stomata responds to that high humidity. So what happens at this point is that the stomata will or how many marks in this question? I would say this question is about 3 marks. 3 marks. Predict 1 mark, explain 2 marks. So to answer this question, so the stomata will take a longer time to close. Will take a longer time to close. So when they take a longer time to close, meaning there will be lesser water vapor diffusing out of the cell. Diffusing out of the stomata, sorry. Diffusing out of the stomata. So what is the effect of there will be lesser water vapor escaping the stomata. So the gut cells, okay, so the gut cells, you refer to the gut cells, will remain turgid. Okay, will remain turgid because no water is leaving the stomata. So no water is leaving the stomata. Therefore, the plant, right? Okay, the plant cell is like this one. Remember the square shape, and yes, the this something like that, and then the nucleus, and then the bunch of chloroplasts. So turgid is when all the pressure on the wall is still there because of water. Water is pushing onto the walls, so it remains turgid. Okay, so there are no water escaping. Where's the vacuole? Oh, vacuole? Oh, this thing lah. This part. But then I would say that should be the vacuole. I'm just sketching it only. I'm just sketching it only. Okay, so this is how it looks like. All right. So do you understand this question? Can we say that there'll be more stomata on the upper surface of leaf as concentration gradient of air is higher? Can we say there's more stomata on the upper surface? As concentration gradient of air is higher. Concentration gradient of air, not specific enough though. And the way be correct to say no, you cannot mention stomata remain open because the moment the stomata remains open, the water vapor can leave. Okay, the water vapor can leave. We're just going through some past year question to relate to this with the transpiration process. Okay, why is the stomata affected? Because the question says stomata. And then transpiration is relating to stomata. Because stomata is basically the door that allows the water vapor to escape. That is the process of transpiration. So when high humidity, what happens is that the outside, the water molecules outside is very, very high. Okay, because humidity means that you can feel that the atmosphere is very wet, that kind of feeling. So outside there's like somehow there's a higher water potential, although it should be a higher concentration gradient in the air, something like that. So because of that, the water cannot leave. All right, the water cannot escape from the stomata. Okay, does this make sense? The vapor doesn't leave. Hmm. The vapor doesn't leave because the water. Yes. Okay. If you mention this, then this is correct. If you mention this, but then you cannot mention water potential gradient. How is the stomata and gut cells when high humidity outside? It depends on, okay, when there's high humidity, right? It means that they will prevent water from escaping. They will try to prevent the water escaping, something like that. Because this is one of the factors that affect transpiration. Factors affecting transpiration. Mm. And you know that in order for transpiration to happen, it needs to have a lower humidity. It means that the atmosphere has to be very, very dry, then transpiration can occur. So, does this make sense? So, the turgid gut cells prevent water. Yes, correct. It's not to say the turgid gut cells, it's that the stomata uh, shuts it. It's basically almost like remain closed so that the gut cells, right, they don't lose the turgidity. They won't lose the turgidity. It's the stomata that creates that barrier, whether it allows or it doesn't allows. Okay. So when lower humidity is just yes, correct. Hmm. All right, this question, this is from one of the questions that I see in the chat. So in the IGCSE biology chat. So how would you answer this question? So this question is saying that describe the pathway of water from outside the root to the xylem vessel at the center of the root. Okay, this one they ask you to use a diagram, but then don't worry, let's just see what is the question trying to tell you. So the question is saying that how does the water move from the outside, means from the root hair cell, from the extracellular environment to inside the xylem vessels. So how would you mention this is by referring to basically this section. All right, so this section is applicable for this question. So how would you explain this for 5 marks? So they say describe only. So we will say that water will enter the root hair cell. So water will enter the root hair cell. And how does water enter? Via osmosis. I'm already getting 2 marks from here already. One is by mentioning that water will enter the root hair cell. This is 1 mark. Via osmosis, another 1 more mark. Okay, then my second point. So why does this happen? You expand from the term osmosis. You will say that the soil has a higher water potential, okay, than the root hair cell. And that justifies the reason why the water will enter the root hair cell. This is my 3rd mark. So I think you can also expand from here. Okay, soil has a higher water, higher water potential in the root hair cell. So therefore, the water will move from higher to lower water potential. So something like that. Okay, then you can also mention that um, it also passes. So since they say use the letters, right? So there will be also 1 mark credited for you to mention the mark basically. Yes, the document will be shared. Okay, so this is the answer. All right. So you know how the marks will look like, right? So 1 mark from here, 1 mark from here. A soil has a higher water potential will also give me 1 more mark, and water will move from a higher to lower water potential, which also gives me another 1 more mark. Okay. All right. So we can write answer in bullet point. Don't write in bullet point, please. Okay. Uh, don't be lazy. Please write in a paragraph form. Okay. Because start now. It's always better to start now. I encourage you guys in exam, please write in full sentences and in paragraph form. Okay, um, it shows that you are writing and doing this exam properly. Okay, all right. So that's it. So Azlan, during paper 6, can you write the planning question? Yes, you can write in bullet points for that paper 6 experiment question. You can write that in the exam in bullet points. All right. Okay, so I'm going to move to chapter 14. So are there any final questions you would like to ask me before I move on? Because we need to get things done. Okay? All right. So this is the one of the questions also found in the Q&A, which is the coordination response, which is relating to the synapses. So I will make this very, very simple for you guys. So, okay, let's take this in this way. I have a presynaptic and a postsynaptic, meaning I assume that the direction is this way. So you follow from left to right hand side. Okay. Yes, I mentioned before that in experiment question you can write them in bullet points. Okay, I've mentioned this already. All right. Okay. So the role of the synapse itself is to make sure that the impulse, okay, like any electrical signals our body has, only can travel in a single direction only. So it cannot go straight and then it go backward. Cannot. It must go straight all the time in one single direction. So we start from here. So in the presynaptic neuron, right, there's a lot of the vesicles. Okay, a lot of the vesicles. So what happens in the vesicles, right, is that inside of the vesicles, it contains a lot of the neurotransmitters which are ready to be released. So when the impulse, okay, imagine there's an impulse coming, okay, it's on its way. It will stimulate the vesicles. They will stimulate the vesicles. And what happens is that after it was stimulated, the vesicles will release the neurotransmitter out to the synaptic cleft. So this is called as the synaptic cleft. Okay, so they throw everything, basically they just vomit everything out into the synaptic cleft, and then what happens is they diffuse, okay, across the gap, and they will attach to this antigen, somewhat like a receptor. Something like an antigen, but not an antigen. Of course, it's a receptor molecule. So these, basically these neurotransmitters, right? They are going to bind the receptor molecule exactly complementary. Okay, so recall back from the enzyme yesterday. So everything, proteins, especially the substrate, must bind exactly complementary. Okay, so once they bind here, then the next impulse is created. So the impulse will be created and it will be going to the next um synapse again. All right, so that's it. Okay, that's it for just this part itself. So okay, from here I explained once already. Any questions? So effect of this is effect of heroin on synapse is removed from syllabus. But then it's removed from syllabus. Okay, molecules bind with receptor proteins on the next neuron. It's the neurotransmitter that bind to with the receptor protein. Okay, then that's correct. Okay. What happens that to the neurotransmitter that don't bind? What happens if the neurotransmitter don't bind? Mm, neurotransmitters, they will degenerate itself. They will degenerate. Basically, they don't work anymore. They just simply don't work lah until, because this can always be produced, unlimited one. So there'll be a lot actually. So there'll be a lot of the antigen receptor, a lot of these receptors. Do we have to know the effect of drugs? No need, no need, no need. What if they ask what's the effect of the receptor being blocked? So if the receptors are unable to, bind, then there will be no second impulse. The impulse cannot be continued to the next one. Okay, because these neurotransmitters are almost like the the key to allow it to continue. So I always think that this is a key, this is a lock. So we always make sure that the neurotransmitter must always bind to the receptor protein so that the next impulse can continue. Without that, there will be no second impulse. Okay. Oh, wait. So the neurotransmitters are produced in the vesicles in the presynapse neuron. Yes, correct. What happens to the vesicles inside the presynaptic neuron again? So the vesicles are almost like a bag, okay, that contains and they can produce a lot of the neurotransmitters. So once that impulse stimulates the vesicles will automatically release the neurotransmitter to the synaptic cleft via diffusion. Why can't I see? Is it like a chain? Chain. What do you mean by a chain? So the vesicles won't be made in the synaptic again. The vesicles won't be made in okay. Assuming that there is still one more, the vesicles will be located here. Something like that. Yeah, help me type down the specific points, okay? Um how does Parkinson disease affect neurons in the brain? They won't ask you how they wait. This kind of question I've seen before in past year paper. Okay, this kind of question is just basically use this synapse concept and apply what happens if Parkinson disease occurs. So there'll be lesser neurotransmitters happening. So there'll be less slower response, slower impulse. And we are in chapter 14. We don't have Parkinson disease, but questions can use an application like a basic situation and test you on it. But the concept is the same thing. The concept is the same thing. So what occurs in the synaptic gap? Synaptic gap. Okay, this right. This is where the neurotransmitter is diffused out. They'll be diffused from the presynapse into the synaptic cleft, and they will diffuse and bind to the antigen to the receptor protein. So whatever J wrote, that one is correct. Okay. So can you give an example of a situation? Yeah, Parkinson disease is one of the situation, but then don't worry, they won't. They will of course tell you the scenarios like this kind of Parkinson disease. They will give you some context on what is it first. Then you only solve the question, all in 4 marks. Yeah, 3 to 4 marks that will work. Mm, it's quite fair amount of marks. But then try to understand this. Okay, so do you get it? Why does this happen? Okay, Parkinson disease, right, is when the body is experiencing a very slow immune response, and it usually happens in elderly people. The hands are shaking all the time. They are shaking all the time. Okay, meaning that they this is involuntary control. It's the body that is automatically doing it. It's not done intentionally. It's that because there are some problems with the neuron, right, that causes the hands to be shaking. That is a Parkinson disease. If I'm not mistaken, this question he sent, right, is from the October-November 2023 paper 4 variant 2, if I'm not mistaken. Mm. So this kind of question, right, they say using the information in figure 3.1 and 3.2 suggest and explain the effect of Parkinson disease on a person's movement. So you see this kind of question, right, is just to apply back your synapse knowledge, which is basically this whole thing that I just explained. Okay? So you talk about that the vesicles are unable to produce it. Yes, what's the role of the mitochondria and the neuron? Is to supply the energy because all these transmission of impulse and everything, they need energy. Okay, they need a little bit of energy. Little bit only. Do we need to know the structure of a neuron? Yes, a big basic one can already. Like, okay, the key structure that you have to know is the motor neuron, the sensory neuron, and the relay neuron. But typically, this kind is just what's the difference between the axon and the dendrite? Okay. If I'm not mistaken, accents is the okay. Accent is something like this. Wait, it looks something like this. I randomly draw. Okay, this is the accent. This wavy, wavy looking shape, looking like branches. These are the dendrites. Okay, this is very simple. What is one thing you think everyone will get it wrong in this topic? Uh, Honestly, if you the hardest questions that possibly it can come out is, of course, one thing is that application question like the Parkinson disease. So that question in the end is not difficult at all. It's just that it wants to test you back on this synapse. You must know what is the question. How does this entire thing work? So can you please give the answer we will write for the Parkinson question for 5 marks? Remember what Jay sent? This one? This. You just modify the answer. Okay? Because Parkinson disease, right? The move the response is lower. So what happens if the response is lower? It means that there are the less neurotransmitter is being released or diffused into the synaptic cleft, and there will be less binding to the receptor. Therefore, the impulse in the post lower, something like that. Correct. Hmm. Can you draw how the sensory motor and neuron? My drawing not very good, but then yeah, sure. So a motor neuron, right? Okay, a motor neuron. If I'm not mistaken, it looks something like this. Wait it looks like branches one, and then it has a. This is a motor neuron. Okay, something like this, and it has a nucleus here. This is a motor neuron. Okay, and a sensory neuron. It also looks similar, but then there's something different. It has this same thing, about the same also, and also like a bit of a same thing like a myelin sheath. Okay, then they have dendrites. Okay, it has also had this small little bulbs. But then the key difference for sensory neuron is has its own this thing. Like a cell body. This is the um sensory neuron. Then we have a last one is the, if I'm not mistaken, is the relay neuron. The relay neuron is yes, it's also in part of the center also, like this, and then the dendrites, and then something like this, like this. Okay. No, I'm not really good at drawing. Okay, but then something like this. So what do these parts? You do not need to know the parts, but all they did is it's just part of their structure that helps to transmit the impulse. Their whole point of having all of these long axons, many dendrites, is like increasing the surface area so that there'll be more impulse able to generate to different parts of the body. I mean to not to different parts of the body, but to other neurons quicker also. Okay. Any questions? Any questions before I proceed? No need, no need. Heroine and the effects, no need. I will just put here because I've been repeating a lot of times. Heroine and effects, no need. Okay, can you go over the eyes? Okay. Well, I need to have the diagram, man. Wait, apologies. Let me go and get the diagram. I don't know whether I have the diagram here or not. I don't think so. No, I do not have the diagram. Okay. Can I go? Oh my god, what to do? You want me to go through the eye, like the labelings, all those? Is it that one? Okay. Okay. Let me have this. Okay, I go back. I go to Discord. Okay, let me go to the chat. Can you see my screen? Can you see? Still can see, right? Okay. So so of course you must know the labelings. All right. The important labeling you have to know is the cornea, okay, which has a C shape, almost like the cortex, C shape. You need to know the iris, the lens, the retina, and the optic nerves. Okay. So what is a cornea? A cornea is very useful for refracting light. Refract, REFRACTS, light. Okay, refracts light, not reflect, refract light. Okay, so and then we have the iris. The iris is the one that helps to control how much of the light is going to enter the eye basically. So it helps to limit it. Okay, and then we also need to know what is the lens. Okay, the lens is basically this one. The looks like a mirror. They help to allow the light to focus. Okay, they make sure the light will directly hit to the retina so that the image can be formed. And the retina is at the back. Okay, near the many of the optic nerves. So they contain a lot of the light receptors, which the rods and the cone. Okay, I'm pretty sure you know what's the rods and the cone. The rods is only to able to see black and white, and the what's that called? The cones is where you can see the color. Okay, all right, and then lastly is the optic nerve. So the optic nerve will carry the impulse to the brain. Okay, so does this make sense? Okay, apologies. My sister is playing some random music. Uh, Please focus here. No worries. Okay, so is is it understood? That's it only. Do we need to know about the aqueous humor? No need. No need aqueous humor and vitreous humor. What you need to know is just the cornea, the iris. Please circle. Please note it down, guys. The cornea, the iris, the lens, the retina, the fovea, optic nerves, and the suspensory ligament. That's it. Oh, I will come to accommodation later on. Okay, hold on, step by step. Okay, all right. Now I will explain the rods and cones graph. Yeah, I need to find. Okay, so this is how it looks like. I just roughly remember what I know. Okay. I use a different pen. Okay, something like that. Okay, so this is the distribution curve of the rods and cones. So this is the rods and this is the cones. So there's always more rods than cone cells. So the cone cells is peak at the middle. So and of course there will be one point. Okay, I should draw it a bit clearer. Okay, wait. Hold on. It should look like this. Okay, so this is how it's supposed to look like. There's a blind spot in the middle. The blind spot is where there is no rods and there's no cones. That's where the presence of the optic nerves is located there. So something like that. Okay, you do not need to know much. You just need to make sure you know what is rods, what's the function of the rods, what is the adaptation of how the rods work and the cones work in our eyes. That's it. Okay, so that's just pretty much what you're supposed to know. Okay, yes. Okay, now we will go to accommodation. All right. So if you take physics, you should know that this is something you have learned in light. You know the convex lens, the concave lens. Okay, so this is about the same thing, and how it all works is because of refraction. Okay, that's why the cornea is its function is to help to refract light. Okay, now you see Okay, as a person, okay, let me see. Okay, now we focus on near point. Near point is where it's looking very close to me. Very close to me now. So what happens is that the ciliary muscles, this is the ciliary muscles, this is the suspensory ligament, it will contract. Okay, the ciliary muscle will begin to contract, and the suspensory ligament will will not be so tight. Okay, it will not be so tight. Okay, because it's almost like the suspensory ligament and the ciliary muscles are working hand in hand. One is contracting, and another one is relaxing, something like that. Okay, so remember, it's always one side that contracts, another side that is relaxing. So for looking closer, see, okay, you try to put your hand close to your eyes. Your eyes will automatically focus. And when it automatically focus, the ciliary muscles will contract. So to pull the focus to the hand that you are looking very near, and they will blur whatever is surrounding it. You get what I mean? So this is where the ciliary muscle begins to contract. Okay. Now, I put my hand further away. Okay, when I put my hand further away, that hand that was far away is the far point. So this is going to happen. The ciliary muscles will begin to relax. Okay, the ciliary muscle will begin to relax. Then the suspensory ligament will tighten so that that distant object is focused. Is focused, and the lens will become longer and thinner at the same time. Longer so it can see further away. Okay? When it's nearer, it will become shorter and also fatter. The lens will become short and also fat. Okay? All right, does this make sense? You really need to use the diagram and also see how you put your hand close to your eyes and your eyes focus on the hand. Everything surrounding is blur. And when you put your hand further away, they will start to unblur the background and start to focus on the hand that is placed very far away from you. Okay? Does this make sense? All good? Any more questions? Any questions? All right, awesome. Okay. What's the function of the fovea? Uh, the fovea is it's just basically the part of the retina where there's a lot of receptor cells which are very tightly packed together. So that fovea is very important so that you can see color vision. You only can see something with color. A lot of the cone cells are located there. So that's why when we have no fovea, we are unable to see color. Okay? So the rod will detect light intensity and cones detect colors. Correct. Mm. So make sense? All right. So in exam, right, for this kind of question, it's never difficult. Trust me, it's never difficult. And every time I try to look back at the hardest question in past year paper, is what makes it hard is because they give you a different situation. The moment they give you a different situation, and you'll be like, what is this? And how am I supposed to solve this? In fact, it's the same thing actually. It's just that you need to think further, think slightly deeper a bit, and then you can definitely find out what are they trying to ask from you. It's always like this. So take some time, take the initiative to think first before you plan out a response. It will never be difficult. Okay? Right. Then any more questions? No, right? Okay. All right. So let's enter hormones. Okay. So for hormones, of course, hormones play a very important role in not just homeostasis itself, but in different situations, hormones helps us to grow and also to help us develop very important characteristics during puberty and all those things. So let's list out the all of these important hormones that you need to know. So the first one is the adrenal glands. And you know from the word adrenal is producing the adrenaline. So adrenaline is very important for the fight and fright. What is it called? Fight and something. What do you guys remember? Fight or something? Ah, fight and flight. Yes. Okay. The fight and flight situation. So this, what happens in the adrenaline production is where the increase in your blood pressure happens, and also your heart rate increases, pupil diameter increases. Okay, so a lot of these can be secret when somebody jump scares you, and then your adrenaline will be quickly secreted out so that you can make the response quicker enough. Okay, so this is very important. The second one is the pancreas. Okay, so the pancreas, they produce the insulin. Okay, and what does insulin do? Is to reduce the concentration of glucose in our blood. We'll explain more this later. Then we have the testes. So the testes will produce the testosterone, and this will cause the develop the male secondary sexual characteristics. Sexual characteristics. Okay, and then we have the ovary. So the ovary will produce the estrogen, and the estrogen will also cause the development of the female secondary sexual characteristics. Okay, the same thing. And last one, from also from the pancreas, is the glucagon. So the glucagon will increase the concentration of glucose in the blood. Okay, does this make sense? Any questions on this? Is ADH part of the syllabus? Nope, it's not. No questions? We will go to menstrual cycle later on. I'm very excited to teach you that later on. ADH is not in the syllabus. No, it's not in the syllabus. Okay, all right. What about pituitary gland? Yes, pituitary gland will secrete the FSH and the LH. Okay, I write that. Okay, pituitary glands is the FSH and the LH. And of course, um, the process of it is by the development of follicles and cause the ovulation. Okay, so that's it. Hypothalamus is the brain control in our brain. Okay, hypothalamus. But don't worry, these are just the important hormones that you need to know. Just know it. Okay. All right, now we enter glucoregulation. So glucoregulation basically means the effect of hormones affecting our blood glucose concentration. Okay, so let's give you a scenario. Okay, this person is drinking two Coca-Cola. So when Coca-Cola is a very high in sugar amount, especially, and of course it's simple carbohydrates, therefore it can cause the spike. Okay, they can cause the spike in the blood glucose concentration. Okay, so how the body is going to do now is by secreting the insulin. So this insulin will convert the glucose. And okay, tell me, where is glucose absorbed in our body? Which part? Where does glucose is absorbed in the body? Glucose liver. liver. I would prefer kidney. Kidney. It's the kidney. Liver undergoes deamination. Hmm. Ah. Yes, the first. You are correct. Yes, I say that where is glucose reabsorbed into. I think it was where is glucose reabsorbed, if I'm not mistaken. Reabsorbed. Mainly reabsorbed. Mainly reabsorbed. Okay, coming back to here. So what happens is that after the glucose has been reabsorbed into our body, the liver, okay, not the liver, the pancreas will begin to detect that high glucose, a spike in the glucose concentration. So they will secrete the insulin. So the when they secrete the insulin, they will convert the glucose to glycogen. Please don't confuse glycogen with glycerol. They are both different things. So convert glucose to glycogen. So glycogen is stored in the body. So therefore, it results the blood glucose concentration to drop. Okay? Yes, it's a negative feedback control mechanism. Correct. So remember, glycogen is a long chain sugar found in animals, usually in animals or in mammals. Okay, glucagon is the secreted hormone in the pancreas. Glycerol is the product of uh, hydrolyzing fats. Okay, make sense? Make sure spelling has to be correct. Okay, so make sure you be careful. Please do not write the wrong term. You definitely minus mark. Deduction of mark will occur if you write something wrong like this. Okay. All right. So now this person lack of glucose now. Okay. So now this person is very tired, and then or maybe is undergoing the strenuous exercise. So he wants more glucose to be sent to the body. So yeah, I'm Chinese by the way. Okay. So what happens is that this person, there of course in the surrounding there will be the low blood glucose concentration. So low blood glucose concentration in the blood in that state. The glycerol is the product of uh, digesting the fats. Okay, so the low blood glucose concentration is detected by the pancreas, and the pancreas will secrete glucagon. So what does this glucagon do is to convert glycogen to glucose, and this results the increase in the blood glucose concentration. Okay, does testosterone affect height, growth, and if so, wait. If so, in what way? Testosterone. Hmm. They do play a little bit role. They do play a small role. Okay. All right. Does this make sense? Do everybody understand this now? Glucose regulation. Awesome, huh? This should be very simple. Okay. Uh. Mm, okay. Tropic responses. Okay. Anything. Should. Which part do you want me to explain? Which part do you want me to explain before I go to tropic responses? in this chapter, in the same chapter. Don't go to the next chapter yet. I say in this chapter first. I explained accommodation already just now. Please check back the recording later on when I explain accommodation again. Okay. Explain response to high and low light. What's that? Response to high and low light. What does this mean? Is it the tropic responses? Okay, never mind. Okay, let's do tropic responses. Okay, so tropic responses is basically it involves with the hormone auxin. Okay, so auxin is the main hormone that is in the for the growth of plant. It's known as a plant hormone. Okay, which allows the shoot to grow. Okay. All right. Okay. So I give you two different conditions. So one has sunlight, another one doesn't have sunlight. Okay. Then I have a shoot. Okay. So initially the shoot is like this, but now I have um another shoot. Okay, I have this one. So where will this shoot go? To the left or to the right? Will it go to the left or go to the right? Correct, left. Okay, never mind. It needs to go to the left. So why is to the left? Is because sunlight. So this process is what we call as phototropism. Photo in the Latin word is called as the light. And you know the source of light is coming from the sun. So this causes the elongation and will go to the based on where the sunlight is located at. Okay, now looking at the second condition. So this is the initial shoot. So what happens to the second one? Will it go to the right, go to the left, or remain the same, or goes down? Elongation means they extend in size. So will it go up or down, left or right? There is no sunlight in this condition. Okay, so it's up. Okay, it can either go up or somehow it may go down. But usually this is rare. This is rare. Okay, it goes down because it's going towards gravity. But of course, the most correct answer is this one, huh? Okay, because assuming there is zero sunlight, so therefore the shoot will be going down, and this is what we call as gravitropism, or some may say geotropism, which is going towards the direction of gravity. So please read the question in the exam. Sometimes they will have given you clue whether this shoot is placed in which condition basically. So this is basically positive geotropism or positive gravitropism. And this one is positive phototropism. Okay, now let's see. How does auxin cause the elongation of the shoot? Okay. So how do you answer this type of question in exam? So the role of the auxin is produced in the shoot's tip. So meaning the shoots like this, right, is produced here. Okay. Then what happens is that the auxin will diffuse down. Okay, they will diffuse down to the shaded region, to the shaded side. Meaning, example here, okay, this is the shaded side. Okay, or maybe it's like this, is here. It will diffuse down here to the shaded region. Okay, and of course, it will be unequally distributed, most importantly. So it will be unequally distributed. But mainly, it's always going to the shaded region of it. So, and then what happens is that this will stimulate cell elongation, which you can also mention by mitosis. Okay, that's it. So this is very simple for tropic responses. So any questions? Okay, so which direction will the plant go? Which one? If there's sun or if there's spinning, okay. So meaning, okay, this question. Thank you. So what happens is that they won't be going to any direction because it's constantly spinning, right? So it means that they do not know, the shoot doesn't know where to go in which direction. Hmm. What kind of experiments cover tropic responses? But the tropic responses are quite obvious. They always will give you the structure. They'll give you the diagram already, and you just basically answer from what you know from there. And the plants are confused indeed. Hmm. What is negative phototropism and geotropism? Negative means they are not going towards light. They are stubborn basically. Okay, basically the sunlight is there. Okay, for example, the light is here, but then instead it goes here. So this is called as negative phototropism. But for negative geotropism, is initially the plant is supposed to go down like this, but then it remains like this. So this plant is being stubborn. So this is called as negative geotropism. But typically, it's either positive or negative lah. Look at the question. The question will always give you the clue whether it's what kind of response that you're supposed to give. Okay. All right. That's it. Okay. This one is positive, right? This is positive phototropism. This one, okay, let me. This is positive. This one is negative phototropism. This one is positive geotropism. This one is negative geotropism. Okay? All right. I'm confused why the oxygen is in the shaded area. Okay. What I can tell you is that um, hormones, right? I would say they are quite sensitive to temperature also. Okay, so basically the auxin, right? They try not to accumulate here because this may cause us the structure to change. So it's always produced here, so it can determine whether it's supposed to go negative or it's supposed to go positive. But then it always depends on the how the stimuli is being provided. Okay, for negative geo, wait, for wait for a moment. Does the position of the auxin change if the plant is negatively phototropic? Yes, they will. They will basically change the position to go to negative side. And for negative phototropism, we move towards the right like a wave. Yes, correct. Hmm. Okay. All right. So, any questions? No, right? Okay. All right. So we come to this another very important part is how do we answer question that is relating to the what do you call this the bacteria. Okay. So we always first refer to so yes, first of all, antibiotic resistant bacteria uses the concept of the natural selection. So it all depends on its surrounding. Okay. So it all always starts off with mutation. Okay, mutation first. So when they mutate, we know it's a change in the sequence of bases in the DNA. When there's a change in the sequence of base in the DNA, what happens is that there will be more variation. So the antibiotic that acts on it, right? Okay, the antibiotic will act by killing the bacteria. That is its main role, correct? Okay, and then what happens is that they will compete. Okay, they will compete for the food space with the mutated. And it's already there's because the one that survives the best will reproduce. Okay, they will reproduce, and bacteria is by binary fission. And then lastly, after successful, the alleles is passed to the offspring lah to reproduce. That's it. Okay. You can you describe binary fission? Fission, not fusion. Fission. So binary fission is the okay for our body cells is by okay, what is the two cell division that happens in our body? What's the two cell division? Please list it for me. Mitosis and meiosis, correct? So bacteria, they cannot undergo mitosis and meiosis because it takes a very a long time. So it must be going through another process because theoretically, if you Google everywhere, bacteria will duplicate in every 20 minutes. So therefore, in order to duplicate in a very high amount, right, it has to undergo binary fission. Fission, so this is the spelling. Binary fission is the replication of bacteria at a very fast rate because it's suitable for them. Bacteria is so small, okay? It's a prokaryote, so they do not have a very complex organelles in their cell. So therefore, they can replicate faster, and they will use this binary fission. What is the allele? Allele is a type of a specific characteristic that that determines one bacteria to another, something like that. So, for example, alleles, right? We may have different allele for eye color, something like that. Okay. So parallel and longitudinal transmission of mutant allele. This is not in the syllabus. Not in the syllabus. Okay. Chapter 16. Oh, your teacher explained. No, it's not in the. It's no need, no need. It's just extra info. It's not in the syllabus. Okay, all right. So we are going to move on to chapter 16. Do you want me to explain plant or you want me to explain this? Menstrual, okay. All right, let's do menstrual cycle. Okay, so this is one of the hot questions that the Cambridge will always ask because that's where students will get confused by which one will produce first, which will produce this, something like that. So we will try to make this very simple is by following my explanation. Okay, okay. So we start from day one. So this female apparently from its previous menstrual cycle, it's unable to have a fertilized ovum. So what happens is that when there is no more fertilization, there will be no implantation immediately. So what happens is that the layer or the uterine lining, the uterine lining, the uterus layer will start to break down. And this is what we call as the menses phase or the menstruation. So that's where a female will, menstruate blood, something like that. So these are all basically the broken down uterus lining. Okay. So what happens is that apart from these 6 days is where the uterine wall begins to break down. Okay, because since there's no implantation, therefore, what is the point of having a thickened uterus when there's no function anymore? Okay, so we look at the hormone level. So within this day 1 to day 6, okay, you can see day 1 to day 6, all the hormones is very very low. Why? Because there's nothing to act on it. There is no any signal or follicle being developed because it's not time yet. So you can see the ovaries, okay, it still remain the same. There's no changes, but then it's ready. It starts to prepare. So okay, after the after 6 days, so what happens is that the uterine lining will start to build up. So which hormone will help with the build up of the endometrium lining? Which hormone? Please answer in the chat for me. Which hormone builds back the uterine lining? Yes, correct. The estrogen. Okay, now we come back. So what happens is that the pituitary glands will start to secrete the FSH. So FSH is the follicle stimulating hormone. From the term FSH, okay, it means that it starts to mature or develop the follicle. Okay, they mature and develop the follicle. This is located inside the ovary. So the follicle is like an egg cell. It's like an egg cell. It's trying to, to develop and mature, something like that. So as the ovary starts to mature, the follicle starts to mature. What happens is that the follicle will start to secrete the estrogen, and this estrogen will stimulate the build up of of this uterine wall. So okay, FSH is produced. Where is the FSH produced? Please tell me which organ produce FSH. Correct, the pituitary gland. So the pituitary gland will secrete the FSH first. So the FSH will act on the follicle by maturing and developing it. And once they develop it already, that follicle will automatically, the pituitary glands in our brain, located in our brain. So this follicle, right, it will automatically secrete the estrogen. The estrogen will start to thicken the uterus lining. Okay? All right, now I'm done with this. So you can see the estrogen level goes up, the FSH level also quite high. Okay, then you'll be wondering uh, later on, now we come to this day 14. So day 14, we always remember is the ovulation time. And this, the pituitary gland will also secrete the luteinizing hormone. The luteinizing hormone will basically ask the ovary to seek to release the ovum out. So it's always the ovary releases the ovum. Please be careful. So estrogen is the one that thickens the uterus lining in the beginning. In the beginning of this menstrual cycle is estrogen. Okay? So this happens. You can see LH peaks. So you know that LH is the one that allows the ovulation to occur. All right. Okay. After this is the ovum. It's now in the oviduct. Oviduct is the same thing as the fallopian tube. So the the ovary will now become a corpus luteum. This is a corpus luteum. Okay. Then what happens? The corpus luteum, after the egg, okay, is being out to the oviduct already, it's ready for fertilization. And it's always the sperm gamete that's ready to fuse it. Okay. Then this corpus luteum at that time will start to release the progesterone. In the first few months, if there is fertilization and there is also implantation, the corpus luteum will keep secreting the progesterone. And what is the role of the progesterone? Please tell me in the chat. What is the role of progesterone? Maintain the uterus lining. Okay, the key thing is to maintain the uterus lining. All right, and then of course they will begin to inhibit the FSH and the LH. Okay, I'm good. I'm good by the way. So it will also inhibit the LH and the FSH. So why is FSH and LH needed to be inhibited? Why? Why it must be inhibited? Both is the same thing. Inhibit and prevent is the same word. So why is FSH and LH inhibited? Yes, correct. So it prevents more ovulation. It prevents the maturing more follicles. Yes, basically to stop ovulation, so that only one egg is able to be implanted in the wall of the uterus later on. Okay, so the corpus luteum in the first few months it will keep on secreting the progesterone, and then what happens is that after the implantation has occurred, the corpus luteum will begin to degenerate, or basically it starts to break down itself. Okay, later on it breaks down. And which part in female reproductive system will maintain the uterus lining? Which part? Yes, the placenta. Okay, so the placenta will basically carry the role of the corpus luteum by secreting the progesterone to keep maintaining it until the fetus is and the baby develop and then it's ready to be released. So corpus luteum first, the first few months will secrete the progesterone first. Then implantation occurs, and after a while the corpus luteum will degenerate, or basically it destroys it or self-destructs itself, and then the placenta takes over by secreting the progesterone. So progesterone can come from two places. First one is the corpus luteum, and the second one is the placenta. But of course, the first one is from the corpus luteum first, because immediately after ovulation, the whole ovary will start to become the corpus luteum. It will secrete it first. Then later on, yes, later on. Corpus luteum. Oh yes, yellow body. Yellow body. Another name is yellow body. So if you write this in exam, it's still acceptable. Yellow body, corpus luteum is the same thing. Okay? We can say again when corpus luteum stops secreting progesterone, meaning okay, corpus okay after ovulation, the egg is released into the oviduct, right? Correct. Okay, so after it's being secreted in the oviduct, the ovaries, okay, basically this thing, after it's been secreted out, right, it degenerates and it becomes a corpus luteum. So this corpus luteum will now secrete the progesterone. They will secrete the progesterone to maintain the uterus lining so that the implantation can occur. After implantation has occurred, the corpus luteum will degenerate after maybe 3 to 4 months. Okay, then what happens is that the placenta takes over, and um, then this happens. Okay, sometimes MCQ question, right? You have to be careful. Careful. Which part secretes progesterone in early pregnancy? So, what's your answer for this question? What's your answer? Which part secretes progesterone in early pregnancy? Early pregnancy. Which part? The corpus luteum or the corpus luteum. If they give you the corpus luteum, then it is the corpus luteum specifically. Okay? No, 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 no. This early pregnancy is just an event. It's just an event. Because the moment the egg is fertilized, right? It's the start of pregnancy already. The moment the egg is fertilized, pregnancy begins there. Okay? So when they say early pregnancy, it's by the corpus luteum. But when it says late pregnancy, it will be the placenta. Get it? All right. We do not need to know the process of pregnancy. But then the question assumes that you understand what does it mean by the start of pregnancy and maybe later on. Later on, late a bit. Okay? Get it? Can you guys finally understand this whole thing? Any questions? Yes, estrogen will eventually stop. Estrogen will eventually stop. You can see the level, it's high, but then it can eventually drop down lah. The main thing for this part lah means the start of the secretory phase, right? Is estrogen. The main protagonist in this part is the progesterone hormone. The estrogen don't really play much of a part anymore. Most of it is always the progesterone. Okay, should I do flowers? Okay, so how are you this good? Uh, I would say when it comes to bio, I try not to memorize a lot. Trust me, I don't memorize a lot in bio. Sometimes I get people saying that biology is a memorizing subject. For IGCSE, it's not. It's a very technical subject, I would say. It's very important that you understand what is happening. Yes, I'm done with my IGCSE already. People, I have a question. Is the grade transfer implemented like the other grade boundaries used for the final grades, for example? No, 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 no. It's the that's the percentage uniform mark. It's just like I would say it's like a percentile, I would say. Hmm. Which chapter should we prioritize? Okay, from my from my advice, okay, I would say is please revise, understand very, very well in the very cold topics. Cold topics meaning plants. Okay? Study the plants. Okay? Know the transpiration, know the fertilization of the plant. Basically, anything related to plant, basically. Focus more on that. Okay? Because questions will somehow come out from there. For May, June now, okay, because last year for my paper, they come out almost a lot of questions is plant related. Okay, all right. Okay, so for fertilization of the plant, right? Okay, it's very simple. So what happens is that okay, the pollen will attach to the stigma. Okay, they will attach here. Okay, sorry, it will attach here. So what happens is that the the there will be a style. Okay, something like that. There's some like small tube. So the pollen will travel down the style, okay, and then it will enter the ovaries. Then it fuses here. See, basically, as being attached, right, the pollen tube will grow. It will grow down the style, and then it will meet with the the ovaries basically. Then fertilization occurs. Yeah, let me see. Okay, I think for this one, I don't know how to explain, but then it's very important that you know that the stigma is the first point, the pollen binds to it, then the pollen tube grows, and the pollen will move down the style, and then it enters the ovary, okay, then it fuses with the ovum, then fertilization occurs. Yes, I will send a PDF later on until everything is done. Okay, all right. We go to protein synthesis, which is one of the hot topics in what you call in this chapter. Okay, so the stigma is just the landing pad for pollen grain, but then it moves down the stem. Yes. So basically, what it does is that this pollination is just to um, transfer it to the stigma, and the whatever happens there is by the process later on already. So it just moves down the style and to the ovary. Correct. Okay. So protein synthesis. So protein synthesis is a very it's frequently asked in exam. So do understand what is happening in the protein synthesis. Okay, so we look at don't look at the others. I just want to explain with a diagram. It's always better. So what happens is that in protein, right, the gene coding, okay, will all happen inside the DNA. Okay, it will remain in the nucleus because the nucleus always contains all of the chromosomes, all of these like very important gene codings are all located in the nucleus. So the gene coding is kept there, first stage. Then what happens is that the mRNA, okay, this mRNA is what we call as a messenger RNA, will carry this copy of the gene. They will mRNA will carry this out, which is coming from the nucleus, and is moved to the cytoplasm. So mRNA is basically made in the DNA basically. Then the mRNA will just basically attach one copy of the gene, which moves out from the DNA to the cytoplasm, and they until they see the ribosome. Then the ribosome will assemble all of the amino acid into a chain. Okay, meaning it's gonna be a chain like this, something like that, a long chain. Basically, the ribosomes helps to arrange the amino acid bases one by one. Okay, one by one, like that. Okay, and then what happens is that the sequence of the protein will be determined, like what kind of protein it will be produced. So the protein is determined by what sequence of the bases is being provided. Okay, so that's it. Are there any questions? I hope you guys are focused, by the way. Please focus, please focus. Can you give a sample question? Okay, trust me, the sample question is just basically describe how a protein is made. Describe how the protein is made. Something like that. 5 marks. Okay, let me just go through briefly one more time. So inside the DNA, all of the gene coding for the production of the protein is inside the nucleus. So all of the coding, all of the bases of the DNA, it's all inside here. And what happens is that this mRNA will undergo this some sort of like, this is what we call as transcription. Transcription is basically like, you know ATCG, right? Okay, you know ATCG? For mRNA, we use AUCG. So they basically transcribe it to for the mRNA to carry. So the mRNA will then copy the gene and move out from the nucleus and to the cytoplasm. Then the mRNA will travel to the ribosome. Then the ribosome will assemble all of the amino acid together to form the molecule. It all depends on the specific order of the amino acids of bases that is provided from the mRNA. Okay? Does this make sense? No need polymerase. This is in A levels. You do not need to mention that. So whatever I just mentioned it, that's it. Any more questions? So does mitochondria have something to do with the process? No. Okay. Wait. Uh, I have a question. How does mRNA move from the nucleus to the cytoplasm? The cytoplasm has cytosol, which increases the acidity of the cytoplasm. Won't wow, you touch in acidity. The the cytosol contains many different kinds of things basically. So it's always been protected. The mRNA is just basically a code, but then it won't be too acidic. Because when it's too acidic, right, the whole cell will just die basically. Wait, DNA unwinds the nucleus. Gene is copied to mRNA. mRNA moves out into cytoplasm. Yes, yeah, that's enough. Correct. This is like chapter. How does mRNA move inside the nucleus? Okay, the DNA, the nucleus, right? They will produce the mRNA. Then the DNA is transcribed to mRNA, and the mRNA is going to take the copy of the gene and move out from the nucleus, then to the cytoplasm. That's it. Okay? Is it better now? All right, excellent. Okay. Okay, we come to chapter 21 already. Okay, so I'm pretty sure that the ecology chapters, there are no problem, right? I think that one is very easy. Okay, so I will go to the important one now. So we come to fermenters. So there are a couple of few questions that came out in about fermenters, but then they are not really very difficult. So the little things that you have to know is fermenters is used typically to produce penicillin. Penicillin is an antibiotic. So it's coming from a fungus called the penicillium. I think you guys know the penicillium. So what happens is that then we have this fermenter. The fermenter is almost like a machine that controls all of the condition to ensure that the result, okay, the product is being produced right, is correctly being made and it's also in the optimal way. So it's also almost like an industrial setting. So we have a probe. Okay, the probe is to measure the temperature and the pH. Is to make sure to check whether the temperature and pH are there any significant changes, because it all has to be under in suitable condition that time. And then we have an air supply. So the air supply contains the different kinds of gases, mainly is oxygen, so that aerobic respiration can occur. Okay, and after that, what happens is that there will be a water cooling jacket. Where's the water cooling jacket? I would say it's something about here, maybe. Okay, so it helps to make sure that the temperature is remain the same temperature. Okay, and then we have a stirrer. The stirrer is to ensure that is everything is equally distributed. All of the nutrients that's being provided, all the things that is inside the fermenter are equally spread out in the fermenter. So all of it are basically, they get it. Do I need to know penicillin? No need, no need, no need, no need. You do not need. Okay. All right. Okay. Then we have this one. This is the genetic. What's the air supply? Oh, to supply oxygen, because it's a fungus. It has to undergo the aerobic respiration. Otherwise, how are they going to grow? Correct. Respiration. Okay. Ah, this is chapter 21. Okay, this is the biotechnology chapter. Yes, I will explain nitrogen cycle. Okay, somebody sent the diagram. Use that diagram. I don't need to go Google and find. Just find me a good diagram. I explain from there. Okay. So how does this genetic modification happens? So first of all, we isolate the human DNA, which is the plasmid. The DNA. Okay, the DNA looks like this. Okay, and we use the restriction enzyme to break this apart. So this point, right, is taken away by the restriction enzyme. And this will form the sticky ends. So this I'm referring to the human gene. Okay, I'm taking that part out. Then on the other side, I have the bacterial plasmid. Oh no. Then I extract, okay, this thing out. Also from the same restriction enzyme. The same restriction enzyme. And I will also leave the sticky end also. It will also be the sticky end. Okay. Then I will use this thing that I just cut off, and I will move it into here. And this will form the recombinant. So this is the recombinant. And what I use to insert that is by what I call as a DNA ligase, which helps to join the cut of the human DNA, and I insert into the plasmid. And then this whole thing, I will insert it back into the bacteria. This whole thing back into the um basically like the fermenter, for example, and let it replicate. Replicate. Okay? Okay, let me check for any question. Any question? Any questions? Wait, because when I was writing, I never see the chat. Please write down any questions. That's it only for the. So basically, it involves genes from different bacteria. Yes, the ligase is a DNA ligase. It helps to join the cut human DNA and join it to the plasmid. So do we use ligase for adding the broken plasmid part into the? No, no, no. We use the DNA ligase to take the broken section of the human DNA and insert into the bacterial one. What we don't use the human. We don't use the human one because the human one will need to undergo mitosis. We don't want that. We want to replicate very, very fast. So that we use binary fission. That's why whatever we do, right, we always insert the human one, the cut human one, into this part. So DNA ligase, I would say, yes, it's almost like an enzyme. It's a ligase enzyme. Mm, okay. So ligase is like the opposite of the not to say opposite, both have different function. Restriction enzyme is to cut it, okay? And the DNA ligase is to connect it back. I would say yes, can opposite. Yes, I think it makes sense if you say this way. Yeah, makes sense if you say this way. Correct, correct. So restriction cuts it, ligase puts it together. Correct. Did you know if you copy word from the MS in your no no what is this? No 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 no. Uh I would this uh I mean we cannot memorize word by word, so we just try to you know just to substitute any word because of course Cambridge won't know whether you are doing that. Are we going to talk about biofuels? Uh Biofuels, there's nothing much about biofuels. Okay, but biofuels is just to it's a renewable energy. First of all, we use yeast that they undergo the anaerobic respiration to produce ethanol, just something like that. So what do we use to insert it? We use the ligase enzyme to glue the cut human DNA to the bacteria one. Okay, all right, that's it. Okay, I need to find the nitrogen thing, the nitrogen cycle. Yes, there'll be a recording of this. Okay, I need to get the. Can somebody send me a diagram of the nitrogen cycle? Can someone do that? Mm. Oh no, why I cannot quit? Okay, finally. What is the difference between genetic engineering and genetic modification? Same thing. Same thing. Genetic engineering and genetic modification is the same thing. Same thing. Okay. All right. All right, let's see this diagram. Okay, trust me, nitrogen cycle is very, very easy. Sometimes I'm a bit confused how you guys are confusing yourself. Okay, let's try not to confuse. Do it one by one, one by one. Okay, so how I always remember this. Okay, this when there's a lightning happen, right? Okay, this lightning happen, we always call this as nitrification. Nitrification occurs. So this lightning will produce the nitric ion. So this is called as nitrification. I hope you know where I'm talking about. Okay, so the nitrification is from the lightning to form the nitrate. Okay, then what happens next? Wow, this diagram is damn confusing. While the arrows are going here and there. Hold up. Okay, I don't know how to start explaining from where. The direction is like so messy. Okay, never mind, never mind. All right, give me a moment now. Let me copy that diagram and I copy paste into my iPad. Okay, hold up. I unshare my screen first. Yes, this diagram is better. Oh my goodness. Hmm. Hold up, give me a moment. Okay, let me share my screen back. Okay, all right. Can you see my screen? Please say yes. Please say yes for me. All right. Okay. Awesome. So let's explain this again. Okay. So lightning. Okay. Lightning. When the lightning strikes onto the soil, they will produce the nitrate ion. So this is the process of what I call as nitrification. So from the word itself, right, nitri means to produce nitrate. Fication is to synthesize it. So I'm producing nitrate ion. So this, I hope, have you guys understand this part? Have you understand this? Nitrification from lightning. From lightning to nitrate ion is nitrification. Okay. All right. Then, so we look from okay, well, it's getting confusing. So nitrogen fixation. Okay, this part. Okay, let's look at this part. Okay, from nitrogen fixing bacteria. So this is the nitrogen that forms into here. Okay, so the nitrogen fixing bacteria, they what they do is that they will they will convert this usable nitrogen so that plants can use it. Okay, so that they will undergo a lot of different process like called symbiosis like that, and then they will do a lot of different things so that this can be used up for the plant. So they will this nitrogen fixing bacteria will convert this into ammonia. Then the ammonia will be used by a nitrifying bacteria. Okay, now I know you guys are confused. What's the difference between a nitrogen fixing bacteria and a nitrifying bacteria? Okay, nitrogen fixing bacteria means nitrogen from the air. Nitrogen from the air. Can you see this direction is going? It's going from the air to the soil. In our atmosphere, we have 78% nitrogen. So this nitrogen will be broken down into a much, like a much soluble form, which is ammonia. Okay? So they will use the nitrogen fixing bacteria first. Okay, then we form the ammonia already. I hope you guys understand from here. So it's just basically from N2 to NH3 for this nitrogen fixing bacteria. All right, then the ammonia that I produce will now undergo by this nitrifying bacteria. So this nitrifying bacteria is another different type of bacteria that converts the nitrogen containing substances into a better nitrogen containing substances for the plant. Okay, how would I say? It's just a better form, a much safer form. Okay, so you want to make this suitable for the plant. Therefore, it has to go in one more time of the conversion with a different bacteria so that it can produce what we call as the nitrate ion. This nitrate ion will only be suitable for the plant to absorb by active transport. Okay, so this will undergo the assimilation. So it's an active uptake of the nitrate ion so the plant so that the plant can use this for their growth. Okay, this is the nitrifying bacteria. So can you now differentiate between nitrogen fixating bacteria and one is a nitrifying bacteria? Yes, all right. So it's don't confuse yourself. Just see this word. One it says it's nitrogen, one is nitrifying. So nitrogen is from is derived from the air. So does the lightning break the nitrogen bond? Wow, this is a very complicated. Lightning is a very strong phenomenon. It has a high electric current. So yes, I assume that it can break the nitrogen bond. Mm. They basically lightning will just they say like from N2 it becomes NO minus 3. So somehow it's oxidized. It's kind of oxidized already. Mm. So um this nitrogen fixing bacteria, I repeat again. So it's taking from the gas from the air, the nitrogen gas from the air, and they convert it to ammonia. Then the nitrifying bacteria will use this ammonia to convert into nitrate ion. Okay, now I make this way. So from N2 gas, I want to make it into ammonia. So I will use nitrogen fixating bacteria. And from NH3, is ammonia. I will convert into NO minus 3, which is nitrate ion. And I will use a nitrifying bacteria. So two different bacteria will do two different things so that they create a better molecule, like a better protein molecule for the plant to absorb. It needs to be safe. Okay, so the bacteria does this job. So how does this bacteria do? Is of course they do by the decomposition now. So they decompose and then to make a different molecule. So yes, so nitrification is lightning from N2 to NO3, correct? And nitrogen fixing bacteria is from N2 to NH3. Perfect. Yes, correct. That's correct. Okay, so now this NO minus 3, it wants to go back to the air. So it has to go by denitrification. So back to the air, N2 is by denitrification. So the okay, so isn't nitrification is just any process that turns something to NO3? Yes, correct. Mm. Uh, yeah, that's correct. Basically, you just nitrify it lah. The whole process is all relating to nitrification and then denitrification. So nitrification is just to keep on converting into a safer form. Okay, but then correct. Yes. So all of this job is done by bacteria. Mm. Okay, so right now, do you understand this? So nitrification is basically using the nitrogen from the air to convert into a safer product for the plant to take up. Because remember, plant cannot inhale nitrogen gas. Gas is a gas, but it needs to be converted into an ion form. Okay, so uh, for us, we eat by proteins, correct? For plants, they cannot eat protein. They only can, you know, uptake those nitrate ions from the soil, and then it's gonna be use it for themselves so that they can grow. Okay? Yes, correct. Okay. So can you delve into the usage of enzyme like pectinase? Just know that you know pectin. Pectin is found in the if I'm not mistaken, is it the skin of the fruit? It's found from fruits. So it just basically convert from pectin to something else one. You don't need to know much. Just just that small thing only. Just briefly know what is it only. I feel more importantly is this nitrogen cycle. I think I've been seeing a lot of people confused, which I don't get it. But then, do you guys understand it now? I think the point is that do you guys don't feel confused anymore, and finally you'll be like, I get what it means now. Okay, so I will send this. Okay, so okay, I repeat the difference again. Okay, so I'll get my teacher to read through the notes. um 1 unit per hour. Don't if you can finish in that one within less than an hour, then you can move on. You don't have to stay purposely 1 unit 1 hour. No, because some chapters like the first chapter, you do not need to take 1 hour. she said I did many past papers, so notes important now. For me, this time is not the right time to do notes. I think you should now just read through your mistakes. That's very important. Read through your mistakes from your past year papers, and then yeah, okay. All right, wait. Let me check. I know somebody wants me to do carbon cycle. Okay, carbon cycle I can remember in my head. Okay, so carbon cycle is all basically the mm, how does carbon dioxide happens to go to the air. So we have okay, I have a tree. Okay, I have a human and I have a factory. Okay, assuming that whatever happens inside here is by combustion reaction. Combustion reaction means it's burning something. So when I'm burning something, I'm releasing carbon dioxide to the air. So this will all go to the atmosphere, CO2. This is the atmosphere. Human will also undergo respiration, which the product is also carbon dioxide. So it also goes to the air. Tree, they also undergo respiration, so they will also go to the air. But one thing is that for plants, this thing can go inside. What is the process that absorbs carbon dioxide? What is the process that absorbs carbon dioxide in carbon cycle? Photosynthesis. Correct. All right. So does this make sense? This is a very simple diagram. So the carbon cycle is all just the circular function of how CO2 is released to the atmosphere, and the only process that can absorb the carbon dioxide is by photosynthesis. That's it. Okay, that's all. So any more questions? No, all right. Okay. So how does greenhouse gas cause increased global temperature? They will they will basically destroy the ozone layer. They will trap the heat. They will trap the heat. Um, then bad things will happen basically from there. Of course, the increase in average temperature is caused by the methane gases. Okay, and then what happens is that the carbon dioxide and methane concentration is very high in the atmosphere. Then this will enhance the greenhouse effect. Then this eventually will cause the climate change. Okay, can you name examples of methane, carbon dioxide, any of the toxic gases basically, but mainly is methane and carbon dioxide. Mm. Okay, can I explain FSH and LH? Okay, I just go through now with you. FSH is to mature and develop the follicle. The follicles are located inside the ovaries. LH is a luteinizing hormone. This allows ovulation to occur. Does this make sense now? Okay, that's good. All right. Okay, so this is the end of. So yes, LH stimulates ovulation. Correct. Okay, so just to sum up everything, yes, I will share this PDF to you guys. Do I mind going genetic modification again? Can you listen to the recording later on once everything is up? I'm very lazy to. Okay. All right. So just the last few words. For mm, okay, one more question I want to ask you guys. Can you guys score 6 out of 6 for your uh planning investigation in paper 6? Can you score 6 out of 6 marks? So anybody struggling with how to write? Yes, I will definitely pin it. Don't worry. Okay. Okay. So how do I remember? Okay. Just remember, I follow this structure. Okay, I don't care, so run away. Okay, so in any question, you just apply this structure. Oh, wait, kind of rude. Wait, what happened? Oh, you mean this one? Sometimes, right? We just my analogy right, are all very um silly, very funny. You know why? Because that what makes me remember it. Okay. So what is I don't care so run away? So I is the IV. You must know what is the IV. Then D, the DV of the experiment. The DV is always roughly is stated in the question. Okay, now I'm explaining the paper 6, the structure of it. Okay, then I have the constant variable. Constant variable always gives two constant variable every time. The moment you give two constant variable, and if it's correct, you're going to get 2 marks straight away for correct constant variable. Then you state one safety. Independent variable though. Yeah, correct. Independent variable. And then we have repeat. If there is a numerical value, like example that this calculation has a lot of different kind of measurements, and then this will be the average. So finding the average of a particular result. CV is the constant variable. Constant variable. DV is the dependent variable. I need help with table design. Can you show how to do 2017 620N? Okay, can you DM me? I will try to do it. Okay. All right. So does this make sense? How to structure your um so constant variable is, for example, right? I'm testing for osmosis now. Okay, where then with potato. So constant variable of the potato is to make sure that the species of the potato remains the same. The size of the potato must remain the same, so that you do not have different kind of sizes. Everything has to be constant, because the IV, right, or the independent variable must be the only one that affects the entire experiment. Okay, so something like this. You get what I mean? So this structure is very helpful. Okay, so 6 marks, very simple to get. State 1 mark for your IV, DV, CV. Give two constant variables, one safety is precaution. Safety is precaution. Control. Oh, okay. Control experiment is basically like, I want to see that, okay, example, if I'm using this particular liquid, right? Okay, I want to see whether if it's with water, will there be any changes? I want to make sure that when I add that particular solution, right, the thing actually changes. That thing actually changes. Yes, I will send the PDF. Okay, I will send the PDF. Don't worry. Okay, the method is to depend on the question. The method, I guess you would know, like, depend on the question. All the question is, they say effect of temperature. If effect of temperature, of course you expand. You know that the constant, the IV, right, is maybe its effect of temperature. How can I maintain the temperature? Okay, how can I maintain the temperature? What is the correct phrase? What is the correct sentence? If I want to make sure my temperature, do not mention water bath only. Thermostatically controlled water bath. Do not put water bath only. So how do I keep the temperature constant? Is by using a thermostatically controlled water bath. Okay? Do not put water bath. Please check back my recording for glomerulus in exclusion. Okay. No, electronic water bath, no. No, no, no. It's the same thing as water bath. Okay? So that's it. All right. So a bit of exam tips. Okay, so I'm pretty sure your first paper is paper 4, right? Paper 4, paper 6, and paper 2, correct? Okay, so for paper 4, um, first thing, it's very unpredictable, first of all. So be prepared for paper 4. Make sure you understand everything. Okay. So make sure you know all of the different processes that are available in the syllabus. Okay. Make sure you be aware of what has been added into the syllabus. So use the syllabus guide to keep on cross-checking whatever learning objective you have missed. Go back to it and read on it. Be prepared. You know, please do not come to the exam and then feel like, oh my god, I miss out on this. Please, 2 to 3 days before, you still have about 25, 26, 27, 28, 29, 30, 1, 2. You got about of 7 to 8 days. So take this time to fill in any gaps. Watch YouTube videos. I don't care whether it's my YouTube channel or other biology YouTube channels. Please make sure you fill your knowledge gaps quickly. Okay? Very importantly, because you need to make sure that you master them. You must master them. Even if you do not understand